You see that mountain over there? Yeah, one of these days I'm going to climb that mountain.
Okay, hello and welcome to the unit, actually the EOC review. This is Mr. Price. You are with second hour, and hopefully they remain somewhat quiet so we can get through this. We are talking about Unit 6 at this point, and where I'd like to start is kind of at the beginning with what you see on, I think it's on your left side. This is the DNA molecule. This formation and this shape is called the double helix. There's some important parts about this that we need to make sure that we understand. This piece that I have in my hand is called a nucleotide. It's got three components to it, or three parts to it. It's got the phosphate part, it's got the deoxyribose part that gives DNA its name, and it's got the nitrogen base part. And if you look here on explorelearning.com, you can see those parts together. You'll see this nucleoside. A nucleotide is adding that phosphate to it. So what I'm building here is a DNA strand, and if I drag my phosphate here, now I can connect one of those deoxyriboses. So if you can see my hand here, that's a deoxyribose connected to phosphate, and then my nitrogen base. So as I keep building this strand, or this single strand of DNA, you'll eventually get to the part where you have a finished product of DNA. Now, this is my original strand, and what I'm going to do is eventually base pair everything. And remember that purines are equal to pyrimidines, and A's and T's always go together. So if I quickly put this together, and cytosine always goes with guanine, so I'm looking for C's and G's. And once I get that put together, I've got my strand of DNA complete, and adenine always goes with thymine here. So again, purines equal pyrimidines, phosphate, sugar backbone, which is this right here, and one single piece of that is called a nucleotide. So those are some of the basics of DNA and the components of it. So as the slow kid here gets done putting this together, what I'm going to be able to do is release this DNA hexacase, which is an enzyme that unzips the hydrogen bonds that go between the DNA molecule. And what I'm looking for is the ability to replicate DNA, or do what's called DNA replication. So I'm going to release that enzyme, and you're seeing this enzyme right here release the hydrogen bonds that bind the adenine and thymine, the cytosine and guanine together. These two now are my original strands, or my leading strands. And what I'm going to do is use DNA polymerase, which is this molecule, basically to go in and make a new molecule of DNA. And notice that my nucleotides are already together, and it's amazing that how long it took some of you guys to do this, and I've done it in less than a couple minutes here while we were doing this lab. So what I've got here is I'm adding A's and T's, C's and G's. I'm making the lagging strand. So this is my new strand, this is my original strand, and of course I've got my original strand over here and my new strand. And what you need to realize is that once we are completely done putting all of these nucleotides together in the right spot, remember cytosine goes with guanine, guanine goes with cytosine, come on nucleotide, there we go. And eventually we get to this part where we have two identical strands of DNA that will form a double helix like you see here, and then we'll go through a process called cell division and mitosis to separate those. So that is DNA replication in a nutshell. Now, switching gears. So I've got a single strand of DNA, and what I want to do, and what DNA is for, is it's basically a molecule that stores information. And that molecule basically gets transcribed and then translated into these things called amino acids. And amino acids are used to make proteins. So eventually all the things that you see in life we get from DNA, going through DNA replication, to transcription, making messenger RNA, and then to translation, which is translating that messenger RNA into amino acid chains which make proteins. And remember, about 4 proteins make up one single red blood cell. So again, we're going to switch gears. RNA, which some of the subtle differences between DNA and RNA. RNA is a single strand. So what I'm going to use is one single original template or leading strand here, and make one strand of messenger RNA. So those same rules apply for, if I have a T, adenine goes with it. So I'm going to put all my adenines in here in my strand. If I have guanine, cytosine goes with it in RNA. And if I have a cytosine, then I've got guanine that goes with it. And eventually what I'm going to get to here is something that is a little bit different and makes messenger RNA unique. And the different base, or the nitrogen base, is uracil. So instead of putting another thymine over here, the green guy, I'm going to put a uracil over here. And remember that right away you should be able to identify RNA for a couple reasons. One, you've got a single strand. Two, you've got uracil in it. And the last part that you should also see, and if you remember right, is you're looking for this start codon, and it's usually the first month that school, and we're starting to think about school starting, it's AUG. So what's going to happen is I just transcribed from DNA this messenger RNA strand, and I'm looking for the first start codon AUG. So again, a codon are nitrogen bases in groups of 3. So AUG is my first, CUG is my second codon, ACC is my third, and then the stop codon in this particular messenger RNA strand is UAG. So basically what I've got to do is match up my codons here, and hopefully I'm smarter than the average kid. I'm going to take these guys and start hooking them together. So, boom. So what this is my anti-codon. So I've got my codon on the messenger RNA, I got my anti-codon on the transfer RNA. So the 3 parts, or the 3 types of RNA are messenger RNA, transfer RNA, and then this purple guy in the background here is ribosomal RNA, or the ribosomes that you can find floating in cytoplasm or on the rough endoplasmic reticulum. So I've just put my anti-codon to my codon. Notice they all match up. This is my first amino acid, which is methionine, is always the first amino acid. The next step, you'll see my ribosome is going to read this and attach, so my ribosome is attaching the amino acids to make my protein. And then what it's going to do is it's going to slide down the, the messenger RNA and keep attaching these amino acids. And this is going to go on for hundreds of thousands and possibly trillions of amino acids to make one protein. Eventually they'll get to this part where it's called a stop codon. Those stop codons will release everything from the messenger RNA and release the ribosome, and it will kick out the amino acid. So ribosome leaves, we have an amino acid which will go in and get bent around and shaped to form things like red blood cells, and this messenger RNA will basically dissolve and start over. Looking over my notes real quick, I want to make sure quickly we go back through some stuff. We have the leading and lagging strands. We have a double helix. We have adenine and thymine go together in DNA. We have cytosine and guanine go together in DNA. Messenger RNA, transfer RNA, ribosomal RNA are the 3 types of RNA. The difference between DNA and RNA, DNA is deoxyribose, double strand. RNA is ribose, a larger sugar, and it's a single strand. I hope that helps, and I'm going to pause this real quick and make sure that I got everything.